genetics, and for good reason. We've mentioned already several times that human genetics are really complicated because there are very few loci that are, there are very few characters that are determined by a single locus. Okay, so human genetics are very complicated. And so this chapter is really about what are these complicated genetic features? What are some things that it's not just like, hey, a little bit beyond Mendel, right? To where it's not just simple two alleles, one is dominant to the other, right? But what are some things that really complicate our study of genetics? First question that we're going to ask to explore this idea is, how does recombination facilitate independent assortment? A couple classes ago, I had you all during our lecture break basically come up with our mechanisms from meiosis that ensure or at least facilitate independent assortment. And one of those is recombination or crossing over. That helps to facilitate independent assortment even when the two loci are on the same chromosome. But we need to talk about how does this actually work, and we also I need to teach you that it's not like we've got these two discrete categories. Independent assortment, not independent assortment. It's a continuum. You have purely independent assortment, then you have things that are mostly independent, all the way down to things that are fully linked, always inherited as a unit. All right? Let's do it. Okay, so the first thing we need to do is we need to set up a two-character cross, right? Which makes sense. If we want to study how two loci are linked, we need to have a two-character cross. We need to look at two different characters, two different loci. And the best way to do this, but you know this already as the budding geneticist that you are, you take two true breeding individuals. So here we've got a true breeding female. She's got the wild type or the dominant allele for both. That's what this plus means. It means wild type, but it's dominant. It is a non-mutant version of this gene. This one without the plus sign, this is not the wild type. This is a mutant version of that gene, which tends to result in a poor protein that doesn't function. Okay? So here we have a wild type or homozygous dominant female for both loci, right? You see that? Both copies of the PR gene are wild type. Both copies of the VG gene are wild type. And then we have a true breeding male that is homozygous recessive for both loci. And you know this, when we do this cross, what is going to be the phenotype of every single individual in F1? Dominant for both, right? So we do this one character cross. Every single F1 individual is heterozygous for both loci. And we call these individuals a dihybrid individual. Heterozygous at this locus, heterozygous at this locus, right? Got wild type from mom, mutant from dad, wild type from mom, mutant from dad. Okay? Keep in mind, these two loci are on the same chromosome, meaning PR and VG, this PR gene and the VG gene, that's not easy to say, and the VG gene, they are on the same chromosome. Okay? So in this individual, if there was no crossing over, you basically have mom's chromosome with the two wild type together, dad's chromosome with the two mutant together. Okay? But if crossing over can, create some independent assortment, then you can get some new combinations. Okay? We okay so far? Yeah, Chris.
The PR and VG, are those like P is one gene, R is one gene?
No. PR is one gene.
Okay.
Yeah,
Right.
PR is one gene, VG is one gene.
Okay.
Yeah. And so you're like, why do they make it so complicated? Can't we just go back to our single letters and capital and lowercase? And the answer is no. No, we can't. I don't know why it's no, but it's no. Okay? And so now typically at this point we're like, cool, we could take two F1 individuals, we can cross them, and then what do we have? What do you get if you cross two F1 individuals? What do we call that cross? A dihybrid cross. Well, we would produce that F2 generation, but that would be a dihybrid cross. And you're like, Dr. Engel, I know exactly what's going to happen when you do a dihybrid cross. We're going to have the phenotypic ratios are going to be what? 9 to 3 to 3 to 1. 9 out of every 16 offspring are going to have the wild type or the dominant for both. 3 are going to have the dominant for the first and the recessive for the second. 3 are going to have the recessive for the first and the dominant for the second. And 1 out of every 16 are going to have the recessive for both. Like, I got that. But that only works if they assort independently. If they're actually linked in some way, you don't quite get those ratios. And here's what we're going to do instead. So what simplifies this, rather than doing a dihybrid cross, is we will cross one of these dihybrid individuals with a homozygous recessive individual. Okay? So we're going to cross one of these dihybrid individuals with a homozygous recessive individual. Okay? Now, the nice thing about doing this cross is you can do the Punnett square and it's not very complicated, because this individual can only make one type of gamete, right?
Yes.
He can only make one type of gamete. And I say he because it's male. He can only make one type of gamete, and that has the mutant version of both genes. He has no other copy of PR except for the mutant copy. No other copy of VG except the mutant copy. Okay? Does it make sense? So you can only make one type of gamete. Now this individual can make how many types of gametes? 4, right? You could have both the wild type. You could have the wild type for here and the mutant here. You can have the mutant here and the wild type here, or you can have both mutants. Okay? 4 different types of gametes. So now when we do this cross, even if you do a Punnett square, you only have 4 columns, 1 row. Whereas if you do a dihybrid cross, you have 4 columns and 4 rows. Right? Okay. So here's the cross. Here's the cross. We take this one gamete that the male is capable of making that has the mutant version of both genes. Okay? Because he's homozygous recessive. That's all he's got. Mutant copies of both genes. This female can make 4 different types of gametes. Okay? And each of these should represent a quarter of all of her gametes. Right? A quarter of all of her gametes should have the wild type for both. A quarter should have the mutant for both. A quarter should have the wild type for the first and the mutant for the second. And a quarter should have a mutant for the first and wild type for the second. Okay? So if they were assorting independently, 25% of all offspring, okay, should have each of these genotypes. Okay? 25% of offspring should end up being themselves a dihybrid individual. You see that? 25% of offspring, which then they're going to show the dominant trait for both. 25% should end up being homozygous recessive. That's just like the original dad was in the parental generation. And then you should have some new unique combos that didn't exist in the cross to begin with, right? Because mom was heterozygous, but look, this is heterozygous at one locus, homozygous recessive at the other. This is heterozygous at this locus, but homozygous recessive at this one. Okay, so these are unique genotypes. Neither parent had these genotypes. Okay, and if they're assorting independently, it should be 25% of all offspring should have each of these. So that's where these expected values are. Expected if they assort independently, 710 for each, because we've got 2,839 total progeny. So it's actually slightly less than 710, like 709 at a fraction, but you can't have a fraction of a Drosophila. Well, you could. You're like, what are you talking about? I tried to pull the adult fly out of the thing when I was transferring the larvae and its head popped off. Isn't that a fraction of a Drosophila? The Monday group, you didn't get to do that yet. It will come. All right. So this is what we expected if they're sorting independently, but that's not what we got. What did we actually get? Very few unique combinations, right? Do you see that? This is a unique combination. Neither mom nor dad had this genotype. Neither mom nor dad had this genotype. These are unique combinations. We call these offspring recombinant offspring, because the only way to get that is to actually have crossing over happen when mom's making gametes to mix the 2 chromosomes that she has. So that's where we get recombinant phenotypes. And then here, these are parental. This is exactly what mom had. This is exactly what dad had. Okay? Exactly what mom had. Exactly what dad had. You see how these are a lot more common than these are? What that tells us is that these two genes are close together. So close, in fact, that crossing over is not always ensuring that they assort independently. Okay? And we can use this to figure out how linked they are. Again, it's not enough to just say linked, not linked. We could do that, but that's boring. We want to know how linked are they, right? I don't want to just know that they're linked. I want to know how linked are they and how commonly does crossing over happen in between them. Okay, so just to sum all this up, you'll remember that recombination happens during prophase 1. And in here, you basically get your one sister chromatid from one of the homologous chromosomes, exchanging some information with one sister chromatid from a separate the other homologous chromosome. And what this does is it creates chromosomes that didn't exist in the parents. Creates chromosomes that didn't exist in the parents. So as long as these loci are far enough apart that crossing over will always happen, then they're going to assort independently. If they're not, then it tells us that they're linked in some way. Yeah.
So if for the flies, going back to the fly example, if they were far apart and not really linked that well, would we expect more of like a 25%?
Yes.
Okay.
Yep. So the goal here is if they're on the same chromosome but they are not linked, if the two genes are on the same chromosome but they are not linked, 50% of the offspring should be recombinant. 50%. And you're like, well, that makes sense because we had two different recombinant genotypes, and each one, you said, Dr. Engel, would be 25% of the offspring. And 25% plus 25% happens to be what? 50%. So if they're not linked, 50% of the offspring should be recombinant, should have a genotype that you didn't find in either parent. If it's anything less than 50%, it tells you that they're linked to some extent. If it is 0% of your offspring are recombinant, are unique combinations that didn't exist in the parent, they are fully linked. They are fully linked. And then so what you would expect would be, man, those things are really close together on that chromosome. If crossing over is never going to mix them, they are really close together.
So, that's if you're seeing for those recombinant, just you have 0 observed.
Correct.
Okay.
Then they are 100% linked. Yep. Anything less than 50% tells you they're linked, and then we can actually get an amount, which we're going to do next. Because we're like, well, I want to quantify this linkage, right? I know how you feel, right? You're like, it's boring to just know that they're linked, and it's boring to just say that there's a continuum, right? And there's more and less linked. That's qualitative. I want something quantitative. I want some numbers. I get it. I'm going to give those to you. Davies.
How would that affect the phenotype of like the offspring?
The offspring? If they're 100% linked? So if they were 100% linked, so we know we went through the effort of initially, when we did our 2-character cross, we went through the effort of making a dihybrid individual, right? Right? Mm-hmm. And they got the wild type alleles from mom, yes? And they got the mutant alleles from their dad. So this dihybrid female, if they were 100% linked, she's got her 2 versions of this chromosome, right? This is the 2 versions of the same chromosome, and those 2 genes are really close together. I want to make it clear. And so we've got the PR gene is here and the VG gene is here. They are really close together on that chromosome. And so crossing over is basically never happening in between those 2 genes. Okay? And so on this one, you'd have wild type. So I'll do that plus sign. So we made this dihybrid female. So on this one, she got the 2 wild type versions of those genes from her mom. And on this one, this is a chromosome she got from her dad and had a mutant copy of both alleles. What that would mean as far as their offspring goes, if these are 100% linked, this female can only make 2 kinds of gametes. She can't make 4 different gametes. right?
Yeah.
She can make gametes that get the maternal chromosome, both wild type, and she can make gametes that get the paternal chromosome, both mutant, maternal, paternal. Okay, but that's it. She can only make 2 types of gametes. She can't make 4 types. The only way she can make 4 types is if you can ensure that these assort independently. Yeah, Chris.
If something's like real, are there like genes that are just not meant to be separated?
Oh yes.
Like,
Yeah, because sometimes you'll get like let's say you have a metabolic process that requires 5 different enzymes, right? One enzyme will do reaction 1, and then a second enzyme does reaction 2. And a lot of times those genes for those enzymes are in the same place on the chromosome. And that ensures that when you are transcribing one of them, you're expressing one of them, you express all of them. Okay? And so yeah, sometimes you do have things where it is very clear that they are not supposed to be separate. Mia, I'm sorry, your hand was up and then I
I was just wondering, like, since like you're testing all this and it's like you're not gonna get like perfect 50%, then like at what point are you like, oh, these are sorting in or not sorting independently? Like,
you could do you'd have to do some statistical test to figure out how likely is it that you got a difference as extreme as yours if they actually are assorting independently. You'd have to use some statistical tool. And the best option would be to use chi-squared, which we're going to do in lab. You're going to love it. Micah.
This is F2, right?
This right here is our F1 female. Now some of the F2 offspring did end up having the same combination, right? But this is our F1 female that we made when we did our 2-character cross of a wild type, true breeding wild type individual with the true breeding mutant individual. Okay, now let's quantify it. You ready? Are you okay? Like the theoretical stuff, is this okay? And I'm not talking about like, do you feel like you're an expert in this, but is it okay? All right. So how can we use this dihybrid test cross to actually map them, to figure out how close they are, to quantify the distance between the genes? Okay, and so here it's basically walking us through the same data we had before, right? Where now it's got, you see the PR and the VG. You see those genes? I know it's hard to see because it's far away for some of you, but I didn't tell you to sit in the back. You told yourself to sit in the back. So here are the 2 genes, same chromosome, yes? Same chromosome? Yes, okay, same chromosome, and then these are the numbers that we had on the previous slide. Now what we would expect is that a quarter of each of our offspring would have these various phenotypes, but that's not what we got, right? These are our observed numbers. So we expected this to be about 710 if you remember, but it's 1,339 of the offspring have the same phenotypes that mom did, same genetic combination that mom did. 1,195 have the same genotype that dad did, and then we've got somewhere around 300 total recombinant offspring that have combinations that weren't present in mom and dad. Okay, so here's how we'll quantify it. What we'll do is this. We will take the total number of recombinants, so all of the offspring that have a unique genotype, a genotype that was not present in mom, was not present in dad, and we will divide that by the total number of offspring. Okay? So total number of recombinant offspring divided by the total number of offspring times 100%, and if they are sorting independently, what should this number be? 50%. Right? If they are sorting independently, 50% of your offspring should have unique combinations. Combinations that weren't found in mom, weren't found in dad. Anything less than 50% tells us what? That they're linked. But now we have a way to quantify the linkage. If this came out to be 0%, they are fully linked. If this came out to be 50%, they are not linked at all. Okay, so anything in between gives us a way to actually quantify how linked they are and gives us a relative distance between the two genes. It's not an absolute distance, but it's a relative distance between 2 genes. So if you ran this calculation for 2 genes and you got this number here, 10% of your offspring are recombinant. You ran this for 2 separate genes and you get 20%. Which two genes are closer together? The one that gave you 10% recombinant offspring, or the two that gave you 20%?
10%.
10%. The smaller the number, the greater the linkage. Right? Is that okay? And so this is all relative distance. Did you have a question, Levi? Oh, okay. Yeah, Mia.
Is it possible for like genes to like separate that should be together, like somehow it just gets split off and then that causes problems?
So like they're so close together that they should? Oh, sure. I mean, anything you can imagine happening to your chromosome, it can happen. It's just a matter of how often it happens.
Is there like an example like where?
Yeah, where you get what we call reciprocal translocation, right? Where you have a piece of chromosomes that aren't homologous but end up exchanging information. And that can happen pretty much anywhere. Yeah. Okay, so here's a map that is put together to show basically the relative distances between genes. And so here's one gene that determines the length of the aristae. And then here's a gene that determines the length of the wings. Here's a gene that determines the color of the body, gene that determines the color of the eyes, gene that determines another degree of the length of the wings, and then another gene to determine the color of the eyes. And so what we're looking at here is for any two genes, we can actually figure out our recombination frequency, and we can get a relative distance between those two genes. So the distance from here to here, the relative distance between these two genes is 13 map units, meaning the recombination frequency between these two genes is 13%. Okay? But then the distance between, say, these two genes, 67. But you can't get a number that high, right? If they're sorting completely independently, what's our number going to be? 50%. So you're like, how on earth do you get this number? How do we get this number? The transitive property, right? Is that the one that it's A plus B? Wait. A plus B plus C. Wait, no, this isn't. But anyways, so if you now I'm all confused. Why do I have to go and start using fancy terms? The distance between A and B plus the distance between B and C should equal the distance between A and C. Right? Does that make sense? Okay? And so that's how we can get numbers like this, because you can't actually get a number higher than 50%. If they're sorting completely independently, that's going to be 50%. You're not going to get more than 50% unique offspring. If you do, that's like a statistical anomaly. There's nothing that ensures that things are going to, favor the recombinant offspring. Okay, so here to here, we can get this number of 13, and this tells us they're pretty close together. Here to here, 48.5%, they're barely linked at all, right? If they weren't linked at all, what would this number be? 50. They're 48.5. That's pretty close to 50. So they have very little linkage between those two genes. And so now we have a way to quantify this, but again, this is telling us relative distance, not absolute distance. Yeah?
So are all of these then the same chromosome?
Yeah, all of these are on the same chromosome. Yep. Yeah, all of these are on the same chromosome. Now, what I want you to do is this. We're going to take ourselves a little lecture break. And I want you to work with those around you. And I want you to come up with a mechanism. I want you to come up with a mechanism that explains why these distances aren't absolute distances. So, in other words, I want you to come up with a mechanism to explain why 13 map units on one chromosome might be different than 13 map units on another chromosome. Okay? Take a couple of minutes, work with those in the group around you, and see if you can come up with a mechanism for that. 2 minutes, starting now.
Because like it's just like the likelihood of during crossover that those are actually so it's just the likelihood of it.
Say for instance, like a centimeter and a half, pushing that on a wire. It might still be 13%.
It's not necessarily the distance in it's not set, it's just like the likelihood of either crossing over that.
That's all I could think
That
of.
they would not sort independently.
That sounds right. We'll go with that. We'll go with that.
Oh, how, what find the mechanism that, I don't understand.
Yes, that's
Like, explain how can there be a difference in how lens, yeah, in the map. Or,
it's a whole 2 minutes and
because we just have a percentage, we don't actually know,
like during the chromosomes, they block out in a mitosis where they would provide the I don't know the last name.
Okay. Do we have an idea on why this isn't an absolute distance, but is rather a relative distance, Tara?
Could it just be from like recombination in the parents?
Okay.
If they weren't linked before and that got recombined somewhere else, it would be even farther apart than the offspring?
Potentially, although the distance between two genes shouldn't change as a result of recombination, because recombination should be basically perfect, right? The same piece of the paternal chromosome switches places with that position on the maternal chromosome. Davies.
I was thinking of mitosis, like during mitosis when the chromosomes are pulled apart. I think that might have an effect on the length of the chromosomes.
Okay.
And like pressing the genes in different locations that cannot be specified, determined, but relatively determined.
Okay. So you're talking about like maybe stretching out a chromosome or compressing it down. Yeah, Autumn.
Do some chromosomes cross over more than others?
They do. And that's the primary mechanism, that the number of recombination events varies from chromosome to chromosome. Yeah, the crossing over is not a random event, right? We talked about this during meiosis a little bit. It is not a random event. It's a very highly controlled, focused event facilitated by molecular machines. And the rate of crossing over, not only does it vary from chromosome to chromosome, but it varies from individual to individual. Some individuals, their crossing over or their recombination frequency is much higher than other individuals of the same species. Another thing, and this is maybe a simpler mechanism to understand, is that when crossing over happens, the chromosomes have already condensed, right? During prophase 1, the chromosomes condense. That's when recombination happens. So the chromosomes are already condensed, and so as you can probably imagine, not every chromosome condenses to the same degree as every other chromosome. It varies from chromosome to chromosome, and it varies even from event, bless you, to event. So, to sum all this up, it's a lot of words, but it's everything we've talked about already. So we cross a dihybrid individual with a homozygous recessive individual. If all of the alleles for each gene assort independently, we're going to generate 4 different phenotypes, right? One that has the wild type for both, which is what our dihybrid individual has. One that has the recessive for both, which is what our true breeding homozygous recessive individual has. And then one that has the dominant or wild type for the first one, mutant for the second, one that has a mutant for the first, wild type for the second. And they should account, each of those should account for 25% of the offspring. The parental phenotypes should be found in half of all of the offspring, should have a phenotype just like mom or a phenotype just like dad. And then half of all offspring should have a phenotype that was not found in mom or dad. And then here's the calculation, here's the equation that we used before to determine a quantification of the distance between two genes. And then here we could put this into an actual number. So map unit or measured in centimorgans one map unit is equal to a 1% recombination frequency. So if your recombination frequency is 10%, how many map units is that? 10. If your recombination frequency is 50%, your genes are fully separate. And how many map units? At least 50. At least 50. Maybe more, but it's at least 50. Micah.
What's a centimorgan?
Centimorgan's just a unit. I mean, it's a unit of relative distance. And so it's a unit that has very little meaning, but it allows you to not represent it in percentages anymore, where you can actually write it out.
It's not related to meters.
No, nope, it's not related to an absolute distance at all. It's named after Thomas Morgan that did all this work with Drosophila. I don't know if he named it after himself or somebody else named it after him. Yeah.
So how do we get like map units that are greater than 50?
You'd have to use genes in between the two. It's like whenever it comes out to 50 map units, you always need to hunt for a gene in between the two to see if it's actually 50 or if it's greater than 50. Right? So if you have two genes, let's say you have this chromosome here, and we have these two genes, we'll call them A and, I don't know, we'll call them B. Okay? And you do your recombination frequency, and it comes out to be 50. Right? Our recombination frequency is 50%, and we're like, oh, cool, these are 50 map units apart. But that may not very well be the case, because even if they were 150 map units apart, what would your recombination frequency be? 50%. And it would come out to 50 centimorgans, right? And so now you have an issue. Whenever you get a measurement of 50, then you're like, okay, we need to find another gene in between here. We'll call it C. And then we can do the distance between A and C, and that comes out, let's say that comes out to 27 centimorgans, and this one comes out to 23, and you're like, oh cool, they actually are exactly 50. Because when it comes out to 50, it could be more than 50.
So let's say, what if C to B was like 43? Would that
C to B is 43?
Yeah.
Oh yeah, so then what we would have here is, what would this be, 70?
Yeah. So we'd find
Yeah.
out it's actually 70 or something
Yep.
like that. Oh, okay.
Yeah, so whenever you get a measurement of 50 centimorgans, you always have to be suspicious that it's greater than 50,
Okay.
right? Because if you get 50, your distance is greater than or equal to 50 centimorgans. You love this, right? I told you. I told you were going to love it, that it was going to satisfy everything you've wanted. I mean, I was just thinking today, but it's probably satisfied everything you've ever wanted, ever. You're welcome. Okay, now we'll get a little bit of human genetics. And the reason why we make such an abrupt transition is we need to talk a little bit about how some features are what we call sex-linked. That the gene that actually codes for whatever that character is found on one of the sex chromosomes. Okay? And so in order to begin this discussion, we need to talk about sex determination in humans, but it happens to be the same way sex is determined in Drosophila as well. So here's sex determination in humans, and this is for all mammals, not just humans. As far as I know, I don't know about the monotremes, like duck-billed platypi. I don't know if sex is determined this way. But so sex is determined in mammals by the presence or absence of the Y chromosome. Okay? Sex is determined in mammals by the presence or absence of the Y chromosome. It's not determined by the number of X chromosomes you have. Now, it is typical for female mammals to have 2 X chromosomes and male mammals to have 1 X chromosomes. 1 X chromosomes. Wow. I'm sorry. That was irresponsible grammatically. I apologize. It's typical for males to have 1 X chromosome, but it is possible that they have 2 X chromosomes and 1 Y. Because sex is determined by the presence or absence of the Y chromosome. Okay? And so, male mammals will always inherit their X chromosome from their mom and their Y chromosome from their dad. Female mammals will always inherit 1 X chromosome from mom and an X chromosome from dad. Actually, that's not always true because you can also have female mammals that have 1 X chromosome and no second sex chromosome. And then they didn't inherit anything from dad, likely, but also could have happened from mom. So unlike most animals, sex is determined by the presence or absence of the Y chromosome in humans, in all mammals, and in Drosophila. Drosophila sex is also determined by the presence or absence of the Y chromosome. So the reason is the Y chromosome contains a gene called the SRY gene that when it starts getting expressed, everything that would be in place to develop the female reproductive system is deteriorated and resorbed, and you generate the male reproductive system. And then when you start to express the sex hormones later in life, you get the secondary characteristics. So in mammals, females are what we call homogametic. They have two versions of the same sex chromosome, and males are heterogametic. They have two different sex chromosomes. Now again, this is for mammals and for Drosophila. Sex is determined differently in other animals. In birds, females are heterogametic and males are homogametic. And then in most reptiles, sex is determined by the temperature at which the embryos develop. Yep. Yeah.
So, is like the Y chromosome, is that like, does it become dominant later, or I mean, how does that work?
Well, there's no corollary. You only have one copy of the SRY gene.
Yeah.
And so there's no like corollary. There's no like a recessive version of that gene. It's just one gene. When you start to express that, it leads to a number of features. It leads to basically the breakdown of what will become the female reproductive tract. I think that's called the Mullerian duct. And then the buildup of the male reproductive duct. And once you start developing the male reproductive duct, you develop the testes. And there are two tissues in there that will, one of them will lead to sperm formation rather than ova, and the other one leads to the production of excess testosterone. So, okay? So, again, everything we talk about from this point forward when we talk about sex-linked genes is going to be true of mammals, and it's also going to be true of Drosophila, because that's how sex is determined in mammals and in Drosophila. This discussion would not be true for reptiles where temperature determines sex, and it would not be true for birds where you have the exact opposite. All right. Yeah.
What's that?
Why is that?
Yeah.
I don't know, but it's very convenient because it allows us to use Drosophila as a model organism. It is very convenient, but why is it that way? I don't know.
Okay.
Yeah, I don't know. I don't think anybody would take this and suggest that we are more closely related to Drosophila than mammals are to any other animal. Yeah.
So then in reptiles, do all the eggs in a single nest often?
No, it tends to be based on how they're buried. So most reptiles will bury their eggs, and the further they are underground, the cooler the temperatures are, and so you usually get a gradient. Now in some reptiles, cool temperatures like sea turtles, and I think most turtles, cool temperatures produce male, and then warmer temperatures produce female. So at the bottom of the clutch, all of the eggs will develop into male, and then at the top of the clutch, all of the eggs will develop into female. But it actually allows you to do some really nice things when you're working with an endangered species, and usually what you need is a lot of females if you're going to try to recover a species, because a single male can father multiple clutches, but a single female can only have a limited number of clutches. So then you can bias the sex ratio entirely towards females if you need more females in a critically endangered species. You just incubate the eggs at the right temperature to develop females. So not all turtles, because snapping turtles, the intermediate temperatures produce males, I think. Really cold temperatures produce females, high temperatures produce females, and then intermediate temperatures produce males. And then crocodilians, it's the opposite. Low temperatures produce females, high temperatures produce males. So, and then some reptiles give live birth, and then they have some other type of sex determination. All right, so what are sex-linked genes? We'll start answering this question by giving you an example. So here's the gene for eye color, and here's normal or the wild type eye color. So Drosophila should have red eyes. Here's the mutant copy of this gene where you can't deposit the right color, and so it ends up being white. And the gene for this is on the X chromosome. Okay, the gene for this is on the X chromosome, and we'll see something that happens with these sex-linked genes. And so here we cross a true breeding red-eyed female with a white-eyed male. Okay, true breeding red-eyed female with a white-eyed male. Notice the female here has 2 of the same chromosome. The male has a different chromosome here. Okay, because the male's only got 1 X chromosome and 1 Y chromosome. And so when you do these crosses, and you do the cross, it's small so you can't see it, but you can look at this later. What you'll find is that when you cross this, all 100% of the female offspring from this cross have red eyes. Some of them are carriers for the white-eyed allele, but all of them have red eyes. And then when you look at the males, half of them have red eyes, half of them have white eyes. Okay? Half of them have red eyes, half of them have white eyes. And so you learn something very quickly about these sex-linked genes, and sex-linked genes in mammals and Drosophila, whatever the trait is for the mutant copy or the recessive allele, whatever that trait is, it's always going to be more common in males than it is in females. Okay? If it's a recessive, because you only have one chance to get it right. You have one version of that gene in males, right? 1 X chromosome, one version of that gene, one opportunity to get it right. And so here's a human example of a sex-linked trait, hemophilia, which this specific mutation is a mutation in one of the proteins in the clotting cascade. So there are about a dozen proteins in the clotting cascade that all have to be activated in the right order to get your blood to clot and bleeding to stop. Okay? And so this is a mutation in the gene for one of those clotting factors, which results in a disease called hemophilia, a bleeding disease. And so here the squares represent males, circles represent females, and then a solid shape is somebody with hemophilia, no color is somebody that's completely normal, and then the partial color is a carrier. And if you look at this pedigree, you'll notice that there's not a single female with this genetic illness. There are females that are carriers. Here's a carrier female. Here's a carrier female. Here's a carrier female. Here's a carrier female. But there's not a single impacted female as far as having the genetic illness. But we have 1, 2, 3, 4, 5, 6, 7, 8, 9 males with the genetic illness. Okay? And again, you have one chance to get a wild type version of that gene. Yeah.
Could this also be like the explanation for color blindness?
Yes, color blindness, red-green color blindness at least, is the gene is on the X chromosome. So it is more common in males than it is in females. At least that version of color blindness. But you still, so if you're female and you're red-green colorblind, you have 2 mutant copies. Both your copies of the gene are mutant. Yeah, Chris.
So did all this start with Victoria, and that's how it got
Yes.
down to?
Yep, the original carrier. And then because of inbreeding, because royal families would rarely, marry with people outside of the family, it leads to an increase in the frequency of rare genetic alleles. Okay. Another good example, calico cats. So here's a calico cat, and the gene that codes for this pigment, either the orange or the black, is on the X chromosome. And so you only have calico cats, you only have female calico cats. You can get male calico cats, but only if the male actually has 2 X chromosomes and 1 Y. And it's very rare. And so wherever you see an orange patch, it's the X chromosome that had the black pigment got shut down. Wherever you see a black patch, the X chromosome that had the orange pigment got shut down. So since male mammals can function with just a single X chromosome, it tells you females need to function with only a single X chromosome. Otherwise, you'd have way too much expression of the genes on that chromosome. Make sense? So in every cell in a female mammal's body, one of the X chromosomes is shut down. and it's not expressed. Okay? And so in here, whichever one gets shut down, the other one gets expressed. And you're like, well, what about the white patches? It's a different mutation altogether where you're unable to deposit pigment. Yeah.
So male mammals definitely use both X and Y chromosomes then?
Yes.
Okay.
Yep. Oh yeah. And then the female mammals use one of their X chromosomes, but every cell uses one, and which one they use is somewhat random, right? One cell might use one X chromosome and one cell might use the other. And this is one of the reasons, it's postulated, this is one of the reasons why autoimmune diseases are more common in females than they are in males, is because in females you're expressing two different X chromosomes. Some of your cells are expressing one of the X chromosome, some of the cells are expressing the other. And so your T cells basically have two different, completely different cell types that it has to recognize as self. And so if it learns to only recognize one of those as self, then every other cell that's expressing the other X chromosome is read as foreign. So, anyways, that's a hypothesis. All right? Any questions? All right. We got through exactly half of the slides, which is good. It's what I wanted to do. And we'll stop here. Have a wonderful day. Those of you that have lab tomorrow, I will see you tomorrow at 11:10. Those of you that do not have lab tomorrow, I will see you 1 week from today. Have a wonderful Easter.